Jesse Swen is from the Department of Clinical Pharmacy and Toxicology at Leiden University Medical Center. And he's, as we just heard, a member of this U-PGx, Ubiquitous Genetics project, and he's going to talk about that. You have to use the mouse if you want to use the pointer.
Well, good afternoon, everyone, and thank you for the introduction. And also thank you for the opportunity to speak to you today about something that I'm really excited about, and that's the Ubiquitous Pharmacogenomics project, or U-PGx in short. And U-PGx is a collaborative effort of institutes in 10 different European countries that received 50 million euro funding through the Horizon 2020 program, as was just explained by Ron. And our objective is to implement preemptive pharmacogenetic testing for a panel of pharmacogenes in a real-world clinical setting across 7 European countries, including Greece, Slovenia, Italy, Spain, the UK, Austria, and the Netherlands. And we will evaluate patient outcome and also cost effectiveness. And the project has actually started on January 1st of this year and will run for 5 more years. And here in the bottom you can see each of the investigators and institutes that are involved. So in recent years we have seen results of a number of randomized controlled trials for a variety of gene-drug combinations that at least to some extent indicate that pharmacogenomic testing prior to drug prescribing is in fact beneficial for the patient. And on top of that, we have seen a steady increase in the number of drug labels that contain pharmacogenomic information that directly impacts patient treatment. And then finally, since 2005, we have the Dutch Pharmacogenetic Working Group guidelines available, and at the moment we have guidelines available for more than 80 gene-drug combinations. However, despite all these developments, application in routine patient care remains very limited. And for that you can, of course, identify many barriers, which are here listed on the slide, and some of them have been addressed and some of them remain to be addressed. But we think that for acceptance it's at least essential that implementation is supported by evidence. So really our project focuses on delivering that evidence. But it's not entirely evidence. It's also to provide hands-on experience to healthcare providers, because we think that also is very important. And in our project, we will address all of these challenges and obstacles for implementation. So to do that, we have designed 11 work packages project that contains 4 different components. And you can see here the first component that focuses on enabling tools. The second is actually the implementation or the clinical trial. The third is more research-oriented. Actually, it's a next step into the future. And then we have a fourth component that's about dissemination and communication and ethical and legal issues. And in my next slides, I would like to dig into a little bit to walk you through this and dig a little bit into the details of each of these components. So the first component, which we are actually working on right now, because it considers enabling preemptive testing. So to be able to do preemptive testing, you need good support by computerized decision support. You need suitable genotyping technology. You need our Dutch guidelines, but you need them translated in your local language, because it's not going to be very helpful to have a Dutch guideline in Italy. And of course, we need to train and educate all involved healthcare personnel. So when I talk about computerized decision support, I already mentioned that we have 7 different countries, 7 different languages. You can imagine that we have more than 7 different computerized decision support systems. So that's going to be a major challenge. And for that, we identified 3 different levels. So the basic level is an interpretive passive system outside the electronic healthcare record. We have the interpretive passive system inside the electronic healthcare record. So there you have the results in the computer, but they don't trigger pop-ups or messages guiding drug prescribing. And then you have the highest level, the interruptive active computerized decision support system, which we have in the Netherlands and which Ron just showed you already. So we will have each of these different situations in different countries, and we are very lucky to have Dr. Matthias Samwald on board of our team. He is working at the Medical University of Vienna, and he has developed sort of a card similar to the one Professor van Schaik just introduced, but this one contains a QR code that you can scan with your smartphone, and then you can have all the recommendations from the Dutch Pharmacogenetics Working Group. And if you want to know more about this, you can go to this website, because I know that Matthias is very keen to collaborate with people who are interested in this. So then the next thing is that you need to have a suitable genotyping technology. And we have allocated budget in the program to buy 7 machines so that each of the sites uses similar technology. And they will all genotype the patient for the panel of so-called pharmacogenes, for which we have a recommendation available in the Dutch guidelines. And so then we need to disseminate those Dutch guidelines. So we published about them in 2008 and 2011. But since then we've moved forward. So we need to translate all the updates into English and then in each of the local languages. And then we will solicit endorsement by other European organizations that are important. And then I will move forward to the second component, which considers the actual trial. And it will be an open randomized crossover trial in 7 countries, including 8,000 patients. And the primary outcome will be a clinical one, and secondary outcomes include process indicator for implementation, cost consequences, and also patient-reported outcomes. So here you can see each of the 7 institutes that are involved. This is actually the Leiden University Medical Center where I work. And what we will do is we will randomize these centers to start with either the U-PGx intervention, and then that will last for 18 months, and then there will be 18 months standard of care. Or you will start with 18 months standard care and then 18 months the U-PGx intervention. And each of the center will be their own control. So this will last 3 years, and then we will have 1 year to collect follow-up data and do the data analysis. So what does the U-PGx intervention look like? Whenever a patient, an adult, is prescribed a drug for which we have a guideline available, he is eligible to enter the trial. He then donates a blood sample, which is genotyped for the entire panel of pharmacogenes, and those results are returned and included in the electronic medical record. And from that point on, we have pharmacogenomics informed drug prescribing. And at set time points, the patient will be contacted to collect outcome data. And if during the period of the trial he receives a second drug, then of course that will also be guided by our guidelines. And there will be also a data collection at set time points, and the same is true for a third or fourth drug. Then what about our endpoint? Our endpoint is on reducing, so the study is powered on reducing the number of adverse drug reactions. So when creating the Dutch Pharmacogenetics guidelines, we classified each of the adverse drug reactions on this scale, which ranges from AA to F, and F is death, that's the worst possible outcome. And we anticipate, this is actually, you can see here the same data. Here you can see the gene, for example, CYP2D6 and the related phenotypes. And here you can see the clinical effect classes that I just showed you. And we have designed and powered the trial to show a reduction in the composite endpoint of the adverse drugs reactions of category C, D, E, and F by 30%. So we anticipate it will go from 4% to 1.8%. Then we know that the current panel-based approach is, of course, limited in many ways. So we have squeezed in a little bit of research also in this implementation project. And for example, we know that 30 to 40% of genetically determined variability in drug PK is confirmed by rare variants that we don't test for. So we will do next generation sequencing for the extreme phenotypes that are included. We also will have a specific look at integrating gene-drug and drug-drug interactions. That was already briefly touched upon. But you can imagine that if you have a drug interaction because you're treated with tamoxifen and you start using paroxetine, so tamoxifen CYP2D6 substrate, paroxetine a CYP2D6 inhibitor, it matters a lot whether or not you're CYP2D6 ultra-rapid metabolizer or a CYP2D6 intermediate metabolizer. And then finally, drug response is the result of the interplay between numerous factors, and we want to do a more advanced network-based approach that's typical for systems pharmacology. So this will be done by the group of Mats Carlsen at Uppsala University, and by doing so we would like to try to improve our ability to predict drug response. So that brings me to my take-home messages. So in the European Union we have large prospective pharmacogenomics implementation programs that are funded and initiated. U-PGx will provide quantitative and qualitative evidence for what we call our new model of pharmacogenomics-guided personalized medicine. And finally, and I think that it's important, we will provide physicians, pharmacists, and nursing staff with the opportunity to gather hands-on experience with pharmacogenetics. And with that, I would like to thank you for your attention. This was our team at the kickoff meeting in January. And if you want to know more, you can email me, of course, or you can look at on our website, www.upgx.eu. Thank you for your attention.